Welcome everyone to my video about biomolecules. Let's start with the explanation of what exactly are biomolecules. A biomolecule is a chemical compound found in living organisms. These include carbon, hydrogen, oxygen, nitrogen, sulfur and phosphorus. These molecules are essentially the building blocks of all of the life forms, and they are necessary for all of the processes that defines a living organism. Now you must be wondering, what exactly are the different types of biomolecules available in the human form? The biomolecules are classified into four different types that are as follows. First of all, there is a carbohydrate. Carbohydrates are essentially the sweets and the sugars that are present in our diet. Chemically they are known as polyhydroxyaldehydes or ketones. They are usually the most abundant biomolecule present on earth and also the most abundant dietary source of energy. Cellulose is a great example. Proteins. Proteins are just as important as they make up around 50% of the cellular dry weight. We usually associate proteins with bodybuilders. However, apart from bodybuilding, protein plays a major role in a lot of other functions. Next is the nucleic acids. Nucleic acids are essentially macromolecules made up of nucleotides, and their main function is to pass on the hereditary characters from parents to offspring. There are two different types of nucleic acids. First, DNA, that is deoxyribonucleic acid, and RNA, which is ribonucleic acid. Lastly, there are lipids. Lipids are a diverse group of organic molecules, and they play a major role in storage of energy in the form of fats, wax, sterol, fat-soluble vitamins, etc. Now, the next part of our presentation will be what are the different roles of these molecules in metabolism. So, let's start with carbohydrates first. Carbohydrates usually serves as a cornerstone in a multitude of metabolic activities. Their main function is the production of energy through the breakdown of glucose into adenosine triphosphate, that is ATP. This is usually done via cellular respiration. ATP is a major fundamental energy currency which is necessary for each and every process that exist in the human form. Beyond this, carbohydrates also play a major role in storing of energy in the form of glycogen and other material. The next is proteins. Their most renowned act is of enzymes because all of the usual biological reactions take a long time. However, due to these proteins present as a catalyst, every chemical reaction is essentially fast forwarded so that it is over in a matter of seconds. Next, they also play a major role in the defense of our body. All of the pathogens that enter our body are usually first encountered by the antibodies, and these antibodies are made up of proteins. Next is nucleic acid. Nucleic acid plays a major role in information storage, as without this information none of the cells or cell organelles might work together. That's why this information is very necessary for the smooth functioning of the cell. These nucleic acids are usually made up of nucleotides, which includes ribose, sugar, phosphate group, etc. And DNA, these nucleic acids also play a major role in DNA fingerprinting. For example, when a particular crime has taken place, using these DNA samples, we could easily identify the person, as DNA is very specific to each and every individual. Next, lastly, the lipids. Lipids are, as I said before, the primary long-term energy storage. While carbohydrates offer quick energy and for a short time, lipids usually store the energy for a long duration in the form of triglycerides. Beyond energy storage, lipids are extremely important as they make up the phospholipid bilayer, which is the cell wall or the cell membrane of each and every cell, which helps the cell to maintain its shape and continue to exist as a cell. And apart from that, let's start to head into the main topic of our presentation, that is the carbohydrates. Now, what are carbohydrates? Well, carbohydrates are essentially a polymer, or rather a long chain made up of recurring monosaccharide units, which are, you know the examples like glucose, lactose, galactose, etc. Now, these monosaccharides, what exactly are they made up of? Now that's what we'll encounter right now. First of all, the functional group present on these monosaccharides are usually the carbonyl group. The carbonyl group, which is the C double bonded to the oxygen, is usually the most reactive part of the monosaccharide. And there are also, apart from the carbonyl group, there are also multiple hydroxyl groups present on this atom. So let's start with the carbonyl group. The carbonyl group could exist as a aldehyde or a ketone depending on the different groups present on it. When an alkyl group and a hydrogen group is present on the carbonyl group, then it is known as an aldehyde, which in turn converts the sugar into something called as aldose. And when the carbonyl group has two alkyl group present on the carbonyl group, then the compound is usually ketone and the sugar is known as ketose. This is a very important part of the carbohydrates, as depending on whether they are aldose and ketose, they are distinguished into two different types, which in further affects the processes that they are involved in. Right. Along with that, the hydroxyl groups present in these molecules changes their name, as they are further known as polyhydroxyaldehydes or polyhydroxyketones. Apart from that, there are also different factors that distinguish these monosaccharides units. For example, the number of carbon present in the carbon chain of the monosaccharides can distinguish them. For example, if there are 3 carbon atoms in the carbon chain of a monosaccharide, then it can be called as triose. In turn, if there are 4 groups of carbon atoms, then it can be called as tetrose. Similarly, pentose. And upon that, there are also different types of structures. For example, some can be open chain, while some can be cyclic in the form of a closed chain. And these also have a different distinguishing point, and they are known as cyclic hemiketal or cyclic hemiacetal. Now you must be wondering that all of these monosaccharides, how exactly do they link together to form a carbohydrate? Now, that is where a phenomenon called the glycosidic linkage comes into place. Here, as you can see as in the example, a glucose and a galactose atom are bonding together. This is done through the beta-1,4 glycosidic bond. Glycosidic linkage occurs through the condensation or dehydration reaction that takes place in between the monosaccharides, and it usually, because of these bonds, all of the simpler monosaccharides come together to form a more complex carbohydrate, which can be a long chain carbohydrate or a short chain carbohydrate. Apart from this, there is also one more distinguishing point between the monosaccharides, that is their stereoisomerism. Due to the presence of chiral carbon, which a chiral carbon is essentially 4 different groups linked on the carbon. When in case of presence of a chiral carbon, there are further divided into dextro and levo. This is also known as handedness, left-handedness or right-handedness. Essentially, in our cell, the enzymes and the various components are very specific when it comes to even the smallest changes in the compound. For example, if you consider a dextro sugar and a levo sugar with the exact same formation in the chemical structure, however, the enzymes would still prefer one of them because of the presence of the chiral carbon. That's how specific the carbons or the enzymes are. Now, you must be wondering, what exactly are the different types of carbohydrates, or rather, what are the different types of saccharides present in our natural environment? So the first of all, we'll start with simple one. The simple ones are further divided into monosaccharides and disaccharides. From the name, you can simply see that monosaccharides is where a single sugar unit is present, and this single sugar unit usually act as a building blocks for all of the other different types of carbohydrates. Right. The general structure of a monosaccharide is usually CH2O multiplied by N, where the N can range from 3 to 7 depending on the compound that is present. Multiple examples are present for monosaccharides which are pretty popular. For example, glucose, which is the primary source of energy for most living organisms, fructose, galactose, ribose and deoxyribose, which are present in the DNA and RNA. Now, we'll head on to disaccharides. According to the name, as you can see, there is a di present in it, which means two monosaccharides units come together through the glycosidic bond to form a compound which consists of two saccharide units. If you can see the photo that I had provided earlier, for example, glucose and galactose, then if you see, there are two units of monosaccharides bonded together through the glycosidic bond. This lactose structure is a great example for a disaccharide unit. And just like that, the example present for these disaccharide units are pretty common. For example, sucrose, lactose, maltose, and lactose is usually found in milk, which is pretty popular for fermentation, lactose fermentation. Apart from this, now we'll head on to complex structures of saccharides. In the complex structure of saccharides, first of all, the one that comes earlier is the oligosaccharides, and the second one is polysaccharides. Oligosaccharides are essentially a smaller version of the polysaccharide, where the units of monosaccharides that are present in the chain is essentially from 3 to 10, which are also linked together through glycosidic bonds. However, they are less common as compared to mono and disaccharide unit, as they aren't available, or rather they aren't as stable in this form. There are two examples, raffinose and stachyose. Now, lastly, the polysaccharides. Polysaccharides are the largest form of monosaccharide chains, and they essentially form the large complex carbohydrates, and they primarily serves as energy storage molecules or structural components in living organisms. There are a large variety of polysaccharides that are present in the environment, and these are the exact molecules that are termed as carbohydrates, while the monosaccharides are just the building blocks of the carbohydrates. The few examples that I have ready are starch, glycogen, cellulose, chitin. All of these are an example of carbohydrates or polysaccharides. Now, till now we have learned about all of the structures, about all of the different types of biomolecules. Now, we'll head on to what exactly are the role of carbohydrates in metabolism. So as you can see, there are three main functions when it comes to metabolism. The first one is ATP production. ATP production is done through the process of glycolysis, and through this process we can get ATP, which is the main source of energy, or rather a currency within the cell's environment. Glycolysis usually takes place in all of the organisms, and some of them might be incomplete while most of it is complete. Now, the second part of the function of carbohydrates in metabolism is lactose fermentation. So, lactose is a type of sugar, as I had mentioned before. It is a disaccharide made up of glucose and galactose. This lactose fermentation takes place in the environment, which we use for our benefits in most of the dairy products. For example, curd, yogurt, everything comes under this lactose fermentation. In this lactose fermentation, the lactose itself is a carbohydrate. So, of course, the carbohydrate plays a major role in the fermentation because it itself is the lactose. Now, apart from that, there is another function of the polysaccharides, that are the biofilm production. Bacteria usually come together and make a safe environment for themselves by forming a biofilm. Now, these biofilms are made up of several other constituents. However, the main constituent and a significant component is the EPS, or rather exopolysaccharide, which could be glucose or other carbohydrate molecules. Why they choose to use this particular molecule? It is because it is sticky, gel-like carbohydrate polymers, which provides the structural scaffold for the biofilm, and it also helps to anchor all of the cells and protect against antibiotics and etc. Now, apart from this, what other roles does the carbohydrates play in cellular processes? Now, in this, as you can see in the diagram, there are two different types of boxes. Essentially, what I am trying to symbolize is that the energy is stored in the form of two different molecules. First one is the glycogen, which usually us humans store in the form of energy in our liver and our muscles, while the other form is usually done by not by humans but rather bacteria in the form of polybeta hydroxybutyrate, which is the PHB. A bunch of bacteria, for example, Bacillus megaterium, Pseudomonas putida, where they usually use the PHB in the form of storage for their energy. PHB, however, isn't exactly a carbohydrate. Rather, carbohydrate acts as a precursor for the formation of PHB. So indirectly, carbohydrates are involved for the process for the formation of PHB itself. Now, apart from metabolism and cellular processes, carbohydrates also play a major role in genetic information, as genetic information is carried out through DNA and RNA, and both of these molecules are made up of a particular ribose group, which is essentially sugar. So this ribose group is one of the main constituent of the formation of DNA and RNA, and this carbohydrates essentially plays a major role, as without this ribose sugar, the entire backbone of the DNA or the RNA would never be formed. So in DNA or deoxyribose nucleic acid, the ribose sugar present is called as 2-deoxyribose sugar, while in RNA, which is the ribonucleic acid, the sugar that is present is known as ribose sugar. Now, reaching towards the end of the presentation, we'll talk about the different techniques used to study carbohydrates and to estimate them essentially. So the first one would be HPLC, or high performance liquid chromatography. HPLC is highly effective for studying carbohydrates, as it is very precise in separating complex mixtures. This process involves injecting a carbohydrate sample into an HPLC, where it is carried by a liquid mobile phase through a column packed with stationary phase. Here, essentially, the carbohydrates are segregated on the basis of polarity, size, charge, and depending on that, the entire solution is separated, and you can identify the individual molecules present in the sample, and you can learn about and study about the particular molecules that you have obtained. The second method that is usually used is the nuclear magnetic resonance spectroscopy, which is the NMR spectroscopy, which works on the fundamental principle of magnetic properties to essentially bifurcate all of the different molecules and study them individually. Apart from that, there is a mass spectroscopy, which is on the basis of mass, and it is elucidated sequences. Apart from that, the last one is the infrared rays spectroscopy, which uses infrared radiation to essentially find out the characteristic frequencies of hydroxyl group and carbon-oxygen bonds, and it is used to study the carbohydrate molecules. You must be wondering, out of all of these four methods, which is the most efficient? And as you can see, I've prepared an increasing order of all of these methods. So the least efficient is the IR method. The second least is the HPLC, then comes the MS, and lastly the NIMR. These are the different methods used to study and the various efficiencies that we have found out. Now, and that was all of my presentation. Thank you for listening, and let's end it.